Okay, hello. Today we are very lucky to have Dr. Marcel Behr with us to discuss his research and his inkling that, or maybe it's more than an inkling, that MAP has something to do with Crohn's disease. MAP is Mycobacterium Avium Paratuberculosis. Marcel Behr is a clinician, a scientist, and professor at McGill University in Montreal in the Department of Medicine and in the departments of Epidemiology, Biostatistics, Microbiology, and Immunology. He's also the founding director of the McGill University International TB Center, and he is now the director of McGill's Interdisciplinary Initiative in Infection and Immunity. Ooh, lots of hats there. So welcome, Marcel.
Thank you for having me. Good morning.
Good morning, and thank you. Thank you, Annabelle, for introducing Dr. Behr. Thank you for being with us, Dr. Behr. So yes, as Annabelle says, you wear a lot of hats. So if you can just tell us a little bit about your background, your research, which focuses on mycobacteria, and you do a lot of work in tuberculosis, but you've also done research in Crohn's and sort of how you became interested in that and what your research interest is. I would love to hear more from you about that.
Briefly, when I was working leading the McGill International TB Center, we realized that on our campus, we had people who were approaching the same disease, TB, from what I consider 4 different strata or 4 different levels. And those are molecules, cells, patients, and populations. And when we brought people in to a room together and talked about the same disease at these different levels, we suddenly realized we all thought we were talking about the same disease, but we were talking about different processes, different research, different angles. And if we got together, there were now some incredible opportunities to build on our different methodologies to make a really fantastic training environment so that a student who's working on a molecule was hearing about somebody else who's working with patients or with communities. And it really strengthened the research community. And one of the outputs is the McGill community wrote the Nature primers on tuberculosis. So we had that kind of bandwidth that we could actually say, this is how you look at it from different angles, and this is how it can come together. And here are the research priorities for the next decade. And that became, in some ways, the template for MI4, which is to say, well, if we have a community of scholars around tuberculosis, we actually also have a community of scholars around HIV. We have a community of scholars around antimicrobial resistance and other themes like that. And if we bring that together, we should be able to find more opportunities for scientists who haven't been colliding to work together and to ask each other questions that strengthens their science. So for instance, one simple example is that in epidemiology, you look at a number of clinical trials and you say something works or it doesn't, and you do a systematic review and a meta-analysis, and that's the way you distill whether there's a signal that a treatment works. Well, you can actually now use that same methodology to look at preclinical research and say, okay, somebody promised that there was a vaccine against, say, COVID in primate models. Well, why don't we do a systematic review and meta-analysis of all the primate models to see if that is promising enough to go into human trials? So sometimes an epidemiologic method can inform how basic scientists work, and sometimes a basic science method can inform how epidemiologists design their next question. And that's what MI4 seeks to do in the McGill ecosystem.
And I think that's incredibly fascinating. And of course, it's MI4 is specific to infection and immunity. And it goes to this theme that we'll talk about with Crohn's disease. You know, it's incredible how many people I talk to that when you talk offline, they say, well, you know, Crohn's might be an infectious disease. And it's not necessarily something they say out loud, but it's something they think and are willing to talk about in a conversation. And so, you know, when you're looking at infection and immunity, what you just said resonates with me, because it seems to me we do have a lot of great Crohn's research happening, but that kind of interconnectedness and the ability to view a problem from multiple different angles is maybe what has been lacking. And your background in infectious disease and mycobacteria specifically, I think is a great example of how we can, bring together sort of different viewpoints to kind of inform how we understand Crohn's. So let's jump into Crohn's and tell us a little bit about how you got interested in Crohn's and some of the research you've done and some of your thoughts about Crohn's disease and mycobacteria.
Sure. So when I think of tuberculosis and leprosy and the community of people who I've been working with, it's been clear all along that these diseases at the end are due to a genetically programmed aberrant immune response to a microbe. That's kind of the standard explanation of TB. It's the standard explanation of leprosy. And if you read a review article of Crohn's disease from the past decade, I'm pretty sure every review article in Crohn's says it's a genetically programmed immune response or immune dysfunction to a microbial trigger. So already I kind of said, well, that sounds kind of similar and it sounds accessible and it sounds like it's part of the, kind of the vocabulary that I understood and the methodologies I understood. So I started off by reading the literature like most of us did. And I found that there were, as I understood, about 3 lines of reasoning that were compatible with a mycobacterium having some sort of a role, either primary, secondary in Crohn's disease. And a 4th that I'm not so sure about. So the 3 lines of investigation that are compatible is when people look for mycobacteria by PCR, they seem to find it more often in Crohn's samples than in controls. And those controls could be either healthy controls, or even if you look at Crohn's, it's more likely than ulcerative colitis. So that would be a disease control. And there's been a number of papers on that, but the methods vary from lab to lab and the details vary from lab to lab. But nonetheless, there seems to be a signal. If you look at the host genetics of Crohn's disease, here there was no pre-selection. People didn't have a candidate. They didn't say, I'm going to look for mycobacteria and see if I find it. They said, we're going to take the whole genome of the human and look at what genes in the human vary in association with Crohn's disease. And the first one was NOD2, which was an intracellular receptor for a bacterial cell wall. Now, I remember being at the medical grand rounds at the Montreal General around 2002 when Jean-Frédéric Colombel announced this in his rounds. And I just thought, this is amazing. I've never heard of intracellular receptors for bacterial cell walls. And I turned to one of the immunologists there and said, is this new? And yeah, these were apparently very new. And the lab of Diana Philpott and Steph Girardin and Gabriel Nunez and a few other people had just discovered these intracellular pattern recognition receptors. So I started to think a little bit more about them and do some research. And in our lab, we can show that NOD2 is a mycobacterial recognition system. And we've done some biochemistry work that says mycobacteria have a different cell wall that is much more recognized by NOD2 than other bacteria. And those are sort of laboratory details. But if you then ask, is it just our lab? No, actually many labs have shown that NOD2 is a mycobacterial recognition receptor. And in humans, variants of NOD2 have been associated with tuberculosis, leprosy, and Buruli ulcer. So it seems to be a mycobacterial genetic recognition system of some sort. So
just to step back for a minute, NOD2, for those who are not familiar with NOD2. So NOD2 is one of the primarily, and correct me if I'm wrong, and there's actually, it's a gene and there are some genetic variants that have been identified to be more prevalent in Crohn's patients. And it seems to be, it was one of the first, I think, that was identified and seems to still be the one that has the strongest signal. Is that a correct explanation, or maybe you can explain it better than I did?
Yeah, thank you for interjecting. I should have defined it better. So when people looked for genetics of Crohn's disease, they found first NOD2 and then a few other genes. And if you have 2 of the risk alleles for NOD2, your risk of Crohn's disease may be 15 to 35-fold higher. So it's a very strong risk factor for Crohn's disease. but it does not explain all of Crohn's disease. And also, you can have these risk alleles and not have Crohn's disease. It does not on its own predict that you will get Crohn's disease. I think some have estimated that maybe 5% of the people at risk will get it. So that means that something like 95% of the people who are genetically at risk are walking around without Crohn's disease. But nonetheless, the investigation suggested that. And then later on, when Judy Cho, who's now in New York, did a larger study of, I think, 150,000 patients and controls and looked at a whole number of genes associated with IBD, she found an association with a whole number of genes associated with mycobacterial infections. So there seems to be an overlap in the host processes that handle mycobacterial infections with the host genes that are associated with being predisposed to Crohn's disease. So the microbe is there, although the methods vary, and the host processes are parallel. And then more recently, I relooked, and I presented this in the meeting in Dublin, at the data on how we give antibiotics to people with Crohn's disease. There was a study by Warwick Selby around 2008, I believe, maybe it was 2007, where they gave antibiotics for a year or not. And they asked if there's a difference between antibiotic and placebo at 16 weeks and a difference at 52 weeks, has the difference increased? And the statistical test said, no, the difference isn't really much better at 52 weeks than it is at 16 weeks. And that was interpreted as a negative study. But there was a difference at 16 weeks, and there was a difference at 52 weeks. And you probably know that yesterday there were these relay races in the World Track and Field Championships in Eugene, Oregon. And if the first person from the relay team is up by 10 meters and everybody else is the same, your team wins. So you still get the gold medal, even if the other people are ordinary or comparable. And so the study didn't ask, well, wait a minute, are we better off at 52 weeks? Because it said, well, because we want to know if the difference grew. If you look from 0 to 52 weeks, the difference is 18%. Now, 18% absolute difference can sound like a success or it can sound like a fail, depending on who you are. If you look at the biologic agents that are used for treating Crohn's disease, and there was a study that Jean-Frédéric Colombel and a few others just published a few weeks ago, and they analyzed the net benefit, so the percent who went into remission, the percent who had a sustained remission for a number of biologics, depending on the drug and the disease. I think it went from 15 to 30% is the net benefit after a year. So the antibiotics are actually not really all that different in terms of net benefit. So I thought, well, that's worth sort of relooking. And then Red Hill did a study that has not been published. And so I looked at their data that they put at clinicaltrials.gov because they haven't published, but they did announce it for filing reasons. And their delta is around 14 or 15% at 1 year. So there seems to be two independent randomized controlled trials that say that being on antibiotics is better than placebo. Now, there's two issues with that. Is 15% a good amount or not? When I treat tuberculosis, I aim for 100% and I expect to have 95% benefit. So I think 15 is a fail for me. On the other hand, before TB was treated, about half of the people got better on their own. So in fact, I'm not getting 100%, I'm getting 50, because 50 might have self-cured on their own. When I treat non-tuberculous mycobacteria, for which we do not have as much literature, and our treatment cocktails are kind of more ad hoc and made up in the lab, we are not up at 95% cure. There's an infection of patients with cystic fibrosis called Mycobacterium abscessus. And there, the sustained cures are around 30 to 50%. So 18% isn't quite 30%, but it's perhaps compatible with what we can get. And the treatments in these trials of Crohn's disease were not optimized. There was there was no preclinical research. Nobody said this antibiotic and this antibiotic and this antibiotic to work the best and reach the best concentrations. It was kind of just something that was made up. At the other end, I was trying to figure out what's another example of a drug and an absolute difference that would just kind of give us kind of a benchmark. And in cardiology for heart failure, the drug Entresto was licensed after one randomized controlled trial where they had a 4.5% absolute difference. And if you read that New England article from around 2014, they said that the trial was stopped early because of an overwhelming benefit. So what I wonder is whether we calibrate success differently in different fields. And in some field, 4.5% is considered an overwhelming benefit. In other fields, 18% is considered disappointing. And I actually don't have the answer to that, but I just think it's interesting that two different randomized controlled trials have found the antibiotic group was doing better than the placebo group after a year.
Yeah, so when you put it all together, so you see sort of the genetic predisposition, which is potentially linked to susceptibility to mycobacterial infection. You see that it presents in some way similarly to what you see with TB and leprosy, which are other mycobacteria. And then you see that antibiotics work. So this is all kind of putting pieces together that say maybe there is an infection here. So then I guess the obvious and maybe difficult question is, why can't we find it? Why can't we find the organism in humans, or can we?
Sure. So I puzzled about that about 15 years ago, and I had a theory back then, and we published it, and it has neither been denied or disproven or confirmed. So back then what we did is we took this organism, Mycobacterium Paratuberculosis, and we put it into mice, and we just harvested their livers and spleens, and we looked for it, and we measured it. And we looked at its length and its width and its shape. And we said it's around 1 micron by 1.5 micron, and it's like egg-shaped. It's not a long stick. And we said, if you look at the bug that causes TB, Mycobacterium tuberculosis, it's about 4 by 1 and it's a stick. So we had this idea that maybe it's not being seen just because it's physically unremarkable. It's kind of like a microbial wallflower. And we said, you need a special lens. You need an oil lens, and you need to have 1,000-fold magnification. So we went and got some tissue from patients. In that case, we got surgical tissue. We went to the colorectal surgeon's office, and we had consent forms. We asked people, you've had surgery in the past. Do you mind if we take out that paraffin-embedded block from surgery in the past, cut one slice and look for it? And they said, sure. We did. They didn't even know that these blocks were just still sitting in drawers. And when we did that, we found these little 1 by 1.5 micron blobs were present in Crohn's, about 8 times more than in controls. We then did a stain looking for mycobacterial RNA, and we found that these little blobs were positive for mycobacterial RNA. And we used a stain that looked for Mycobacterium avium, or a stain that looked for Mycobacterium tuberculosis. And if it was M. avium, it was positive, and it was M. tuberculosis was negative. So I thought that was kind of possible, that the reason we hadn't found them is they were small and people had been using the wrong microscope. That was published, I think, in 2007. And in the subsequent 15 years, I don't think anybody has either tried to prove or disprove it. And maybe I'll have to do it again, but it's not independent proof if you do the same study yourself again. Other people have hypothesized that the mycobacteria are there, but they shed their cell wall. They take off their clothes and they end up in a different form or a different shape. That's a possibility also, but that makes it harder to look for them, because then you have to develop brand new tests and brand new assays, and you have to validate them in other ways. And I've had difficulty demonstrating that. And when we did our mouse experiments, when we stained for the wall or when we stained for the DNA, we saw the same number of bacteria. So at least in our mouse experiments, there was no evidence that they had lost their cell wall.
So what's interesting to me about MAP is that, so we know it causes a disease that presents similarly to Crohn's disease in cattle, correct? And it's called Johne's disease. And we know it's the causative organism of Johne's disease, and we can harvest it. We can identify it in cattle. So there seems to be, at least theoretically, if the organism is relevant in Crohn's disease, some difference between cattle and humans, where you can readily test for it in cattle. And we know it's in cattle. We know it's very prevalent in cattle. And, you know, it may be that we're consuming it when we consume dairy products and other things, but we can't find it in humans. And this has always sort of puzzled me about this whole MAP theory, which has now been out there for a century. It's an old theory. It's not a new theory of Crohn's disease. It may, in fact, I think, be the original theory of the cause of Crohn's disease, or at least a subset of Crohn's disease. And 100 years later, we still don't have a validated human test for MAP, correct?
Sure. So there's two parts to that. So earlier I said there were three reasons to think there might be a link, which was microbiology, immunology, and pharmacology. Previously, people have said the other reason for there's a link is analogy, that the same bug already causes a similar disease. I'm not a pathologist, so I'm actually not in a position to say there is or is not strong analogy. And I've tried to read that literature to the extent that I recognize it, but I'm not actually sure they are that similar. They may be similar, they may not be similar. And I would be very interested to know if we had a gastroenterologist do a colonoscopy on a cow with Johne's disease, if they say, wow, this looks the same, or they say, this isn't the same, this looks like C. difficile, or this looks like Shigella. So those sorts of exchange experiments I think would be very enriching. Having a sheep pathologist looking at Crohn's tissue, having a Crohn's pathologist looking at Johne's tissue. And I'm not sure how well that has been done, honestly. So that's the first part. But the next part to your question is indeed spot on, is when people have looked for mycobacteria in human tissue, they say, oh, mycobacteria are fragile, they're hard to work with, they're difficult, they're fastidious, you have to, talk with them in a nice voice, and you have to put on the mood lighting and all this type of stuff. But on the farms of America, you can grow MAP out of the manure behind the cow. You don't even test a cow, you test a herd. And to test a herd, you just look at the feces. And if the manure is positive, it means one of the cows is positive, which means the herd is positive. So it does not appear to be a difficult organism to grow in its natural environment. So invoking that it's difficult to grow in humans is a little bit of a stretch to me, and I concede that point.
And you talked about analogy. The other analogy that I think is interesting is the analogy with intestinal tuberculosis, which, as I understand it, looks an awful lot like Crohn's disease as well. Is that right?
Yes. Again, I haven't done side-by-side histopathology, so I think there's overlap, but some distinctions. But on a clinical profile or clinical portrait point of view, I was taught in my infectious disease residency that Crohn's disease in somebody from a TB endemic country is a TB diagnosis until excluded. And that is still largely the way we practice medicine, is if somebody presents as having Crohn's-like disease and we don't know, and they haven't had a biopsy, they haven't had pathology, they haven't got a confirmed diagnosis, but they have a TB contact, we will aggressively do biopsies and look for a culture for Mycobacterium tuberculosis. There is enough of an overlap, at least clinically, that you can't tell between the two until you've done thorough testing.
And are there any biomarkers for infection in a human of mycobacteria, of mycobacterial infection? Are there any existing biomarkers?
Yeah, so there's two tests that are used in tuberculosis. One, obviously you look for the microbe, but the biomarkers would be things like skin tests, like the tuberculin skin test, or the quantiferon test to look for an immune response specific to mycobacteria. The quantiferon test looks for proteins that are made by Mycobacterium tuberculosis, but not by Mycobacterium avium. So it's deliberately made to be specific, and therefore it would deliberately be useless to look for Mycobacterium avium because it avoids the contamination of Mycobacterium avium in the test. So that's one type of a biomarker. We call that immunodiagnosis. And that's, if you like, clinically validated or FDA approved tests. And then there's a whole bunch of research in the tuberculosis field to look at other biomarkers, which we would call immune sort of like signatures, RNA signatures, or things like that, that are asking of all the people in the world who have a positive skin test, or all the people in the world who have a positive quantiferon, which one is likely to develop TB in the next 3 to 6 months? And so these are tests looking for what's now called incipient tuberculosis, or asking if you treat somebody for tuberculosis, do these signatures go away? And it tells you your treatment is doing well. And there's a lot of action on the research forum there, but there's nothing that you would call currently an FDA approved test that we are now applying in our clinics.
Okay, and going back to the discussion of antibiotics and this notion that based on the two trials that you discussed, the Selby trial and the Red Hill trial, that it seems to have some net benefit over placebo. Is it possible that, again, I think you made the point about not optimizing the therapy. Is it possible that if there is an infection, it's just difficult to eradicate? We may not have the right combination of antibiotics. How do we sort of tease out why antibiotics work? Like, are they killing something? Is it some kind of anti-inflammatory benefit? Is it just that they're reducing kind of the load in the bacterial load in the microbiome, and so there's less of an immune response? Like, how can we figure out the mechanism of action about why antibiotics work? And then as a second step, look at, all right, well, if they work, let's optimize them to that particular mechanism of action.
Yeah. So, as an infectious disease doctor, part of my business is giving antibiotics. It turns out a bigger part of my business is stopping antibiotics because they're used and overused. When we give antibiotics in some diseases, we try to kill bugs dead. It's like those insect killers on television commercials. And so in some cases, like if you have a bloodstream infection on a heart valve, the goal is to kill every last strep viridans that's on your heart valve so that you do not have a relapse of that infection. In other infectious diseases or other applications, if you like, of antibiotics, we're trying to beat down the number of bacteria so that the host can then get control of it. And that's probably what we do with the pneumonia, is your classic streptococcal pneumonia, strep pneumonia. You give antibiotics, and after 2 to 3 days, people feel better. We may not have sterilized every last strep pneumo in the body, but by then we make antibodies and we probably clear it. And so in that case, our goal is not to kill bugs dead. Our goal is to help people. And then at the most extreme example, if you have chronic lung disease called bronchitis or chronic obstructive lung disease or bronchiectasis, there's been a number of trials that say that when you have an exacerbation, so you're coughing, you're producing more spit, and you're having difficulty breathing, whether you give amoxil, doxycycline, or Septra, all groups are better than placebo. And there you're probably just saying the person may have a viral exacerbation of their bronchitis, because the viral exacerbation, instead of having 10 to the X bacteria in their lungs, maybe now they have 10 to the X plus 1 or 10 to the X plus 2. They have 10 or 100 times more bacteria. and we're just beating it back to a tolerable level. So when we give antibiotics to people with Crohn's, we should be thinking about, are we trying to sterilize something? Are we trying to reduce something to a level the immune response gets rid of? Are we trying to reduce something to a tolerable amount so that we don't care anymore? And I'm not sure if we know the answer. And you use the word mechanism, Ildiko, and I'm not sure we need to go for a mechanism. I've wondered for a long time about, understanding what's the mechanism or the etiology, and therefore coming up with a treatment, improving it that meets the mechanism. And yet I've also been wondering whether we should just be asking if antibiotics make people better, and if they make people better as well as Humira makes people better, is that not a desirable goal on its own? And if you first show things make people better, you can maybe worry about mechanism later. And maybe that's more pragmatic, but at least antibiotics have a very well-known and defined safety profile. And if you give antibiotics for a year, we generally don't think that you have risk of things happening 10, 20 years later. So there are two kind of polar ways of thinking about it. How do they work, or do they work?
No, I think you make an excellent point. I think that's spot on. And if you look at the vast majority of our FDA or Health Canada or EMA approved drugs, we don't know the mechanism of action of actually most of them. Yeah. We just know that they work. And I think that's true for many of the biologics too. We have some idea of the mechanism, but not actually a very robust understanding there either. So I think your point is very well taken. If we know they work, let's use it. And I think there's an element of maybe antibiotics are not as sexy as some of the new recently approved FDA drugs, but I think it is a very valid question to ask, but do they work just as well? And I believe antibiotics are used, correct me if I'm wrong, for post-operative recurrence of Crohn's disease and pouchitis and some other things like that. So it is used, it's just not used really as a primary treatment in Crohn's. Is that right?
Yeah, that's my understanding. I'm not a gastroenterologist, but from what I've seen in clinics, when people have an exacerbation, they could be getting 2 weeks of ciprofloxacin and metronidazole commonly. But again, what the antibiotics are targeting would not be known at that point.
Like, I've always wondered, and maybe this is just my crazy hypothesis, but there's a lot of talk in the GI community, at least my understanding of it as an outsider, as a layperson, a lot of talk about early aggressive therapy. But I think of that in a slightly different context than how it's used. What they mean is start an anti-TNF very quickly. What I think is, okay, you have a patient present with Crohn's, they're diagnosed with Crohn's. What if you sort of immediately, yes, maybe started an anti-TNF, but also started antibiotics, also maybe put them on dietary therapy, whether that's EN or some other type of dietary therapy that has also, just as good of a response rate as many of the drugs do. What if you sort of did an everything in the kitchen sink approach right at the initiation of the disease, or at least at diagnosis? Would you have better outcomes? That's just something that's always been kind of at the back of my mind. I'm wondering if you have any thoughts about that, about early intervention with antibiotics alongside something like an anti-TNF.
I've wondered about it. I can't say that I know that it would be better or it would be worse. We also have, by analogy in clinical medicine, if somebody has lung disease and they come in with an exacerbation and they have heart disease, then you have somebody who gets put on steroids and puffers and diuretics and 2 antibiotics. And you say, well, we've thrown the kitchen sink at them, and that should be better. And it could be better, and they could be better 24 or 48 hours later. But the other issue is if you give somebody 6 or 7 drugs, and 2 days later they have an arrhythmia or they have a rash, then you're stuck going, well, they're on 7 drugs and they have a rash, which one caused it? Or they're on 7 drugs, they have an arrhythmia, which one caused it? So I think you'd have to be cautious about thinking about the incremental benefit of everything you add and the potential for incremental harm of everything you add. And when you add things together, you also have drug-drug interactions, as you're well aware. So, as one example, when you treat people with tuberculosis and AIDS, it's important to treat the TB and then to get the AIDS drugs going as soon as possible. And there's good randomized control trials saying that you should not treat TB for 6 months and start treating the AIDS after 6 months. So good, start earlier. But does that mean you should be doing everything on day 1? Or can you treat 1 and then see if things are stable? And then if the tuberculosis involves tuberculosis meningitis, which is inflammation of the brain, is it a good idea to add the AIDS drugs immediately, or should you maybe wait a week or 2 weeks? Or should you add the AIDS drugs and the TB drugs, but now you have to add steroids to reduce the inflammation? So there are other examples in medicine where we try to treat multi-pronged, but each time you have to think about how the different elements can be mutually beneficial or can be potentially antagonistic. How this would work in Crohn's, I really don't know. Some might say, well, why don't you try antibiotics first? And if they don't work, try biologics. Others would say, well, why don't you try the standard of care biologics? And if they don't work, try antibiotics. I see no harm, of course, in dietary modification, but I think dietary controlled studies, I think, are very difficult to do, if I understand. But you probably know much more about dietary modification studies than I do.
I have a question about the antibiotics. There are a lot of people on the there are people who are on the triple antibiotic therapy, but they're not on it for just a year. They're on it for years. Do you see any problem with that? And also, one of the antibiotics used is supposed to go after dormant cells. Is that something that you see in treating TB patients? Do you do that as well?
Yeah. So the duration therapy is really unknown. When I treat something like pulmonary Mycobacterium avium, I usually expect to know within about 4 months if the person's getting better. We would repeat CAT scans and we would repeat cultures. And then we would, once the cultures are negative, we treat for 12 more months. So that's a standard of care for a pulmonary Mycobacterium infection. So it could be 4 plus 12 months or 6 plus 12 months. And we have no evidence that going longer than that is better. There, of course, are people who can have a Mycobacterium avium infection in 2017, who can have another Mycobacterium avium infection in 2022, but that would not be a relapse. If somebody goes from culture positive to culture negative and is fine for a few years and is culture positive again, that's usually a reinfection rather than a relapse. Regarding dormant bacteria, that's been a separate controversy in the TB literature. And I've recently learned that if you go to Wikipedia and you look at latent TB, there's a section called controversies. And in that section called controversies, somebody says that I wrote a controversial article about latent TB and whether there are dormant bacteria in 1/3 of the world's population. So that's an ongoing debate in the TB community as well. I think that there are 2 types of dormancy that are being discussed in the TB research community. One is whether there is a biologic dormancy in people who've been infected who haven't had disease, which is just happening naturally. And the other is whether when you give antibiotics, the antibiotics are exerting pressure on the bacteria to go into some sort of a non-heritable, persistent mechanism that's harder to cure. And there's a lot of fantastic work by some scientists, especially in New York City, on trying to understand how to kill that last bacteria. If the first 2 antibiotics are sort of slowing it down, you might need a different strategy with the 3rd antibiotic. But that hasn't been the science is fantastic, the biology is fantastic, but it hasn't led, to my knowledge, to a product that we use in the clinic to target that dormant bacteria.
I want to ask you about the BCG vaccine, and this is probably, again, just theorizing, hypothesizing. Do you think there might be any role for a preventative benefit of BCG vaccine for Crohn's disease?
Is that the tuberculosis vaccine? Okay.
Yeah. So BCG is given in many parts of the world, probably 100 or 120 million doses a year, and it's given mostly to newborns. And the greatest benefit has been seen in preventing infantile forms of TB. So meningitis or disseminated TB in the first few months or years of life. And even in Canada today, in 2022, we give it in villages that have a high incidence of TB to protect newborns. Whether BCG provides or confers any protection beyond 3, 5, 7 years when it's given at birth is poorly understood, but we know that BCG converts your tuberculin skin test. That's the thing that they put a needle in your arm. And if you give BCG at birth, the tuberculin skin test will come back to normal by about age 10. So that suggests that BCG does not have a more durable effect than maybe 5 or 10 years later. So if BCG is given at birth and people have a MAP infection at 2 weeks or at 4 weeks, maybe it would be protective. If BCG is given at birth and somebody has a MAP infection at age 15, it's unlikely to be protective. And so showing a link between BCG and Crohn's disease would be very difficult. It would be a time dependent. And we've already discussed how we don't have good tests for showing people have a MAP infection. We can't even know if people are getting MAP infection 1 per million per year. These are sporadic things like food poisoning, or if this is a more common thing that's like, say, salmonella in chicken, that we're often being exposed to salmonella every day, or depending on how you prepare your meals and stuff. So we don't really have a good handle on whether people are getting infected with MAP commonly, and if so, at what age. And therefore, it's hard for me to know if BCG could or could not be of any benefit.
And you made a comment earlier, which sparked a question in me too. You know, we know that Crohn's patients have flares, right? They'll have a flare, they'll get a treatment, they'll go into sometimes long-term sustained remission, they have scopes, everything looks great. And then several years later, they might have another flare. I've often wondered, is that just a reinfection? Like, did they maybe clear an infection and they were all right for a period of time, or maybe it was the therapy that they were on. And then at some point, some years later, there was a reinfection with whatever the causative pathogen might be.
It's a possibility. We don't really know the cause of Crohn's disease, but we also know that other intercurrent illnesses can affect how our host response evolves over time. So if you take the tuberculin skin test, where you put proteins of tuberculosis under the skin, you look for an immune response, you think, okay, that's specific to how you respond to that. But it was known back around 1907 or so, I think it was von Purke or Montu who showed that if you have measles, your ability to respond to the tuberculin goes down. So you can invoke, oh, I have an infection, I have a flare, therefore I got reinfected. Or you could invoke, I had an infection, my body had created a stalemate with the infection, and then I got something else. I got norovirus on that cruise, or I got a measles vaccine, or I got my 3rd dose of my Moderna, and that triggered some sort of a difference in my immune response to how it was handling something else. So I can't really know between those two what might or might not be happening.
No, that's a really, really great and fascinating point. Absolutely. Absolutely. And you know, we digress a little bit, but I find the recent research on the connection between the Epstein-Barr virus and multiple sclerosis, that they're now saying it is causal. Maybe that's that same thing that you've just described as well, which is really interesting. Two other quick questions. One of them is about phages as a potential therapy. Like if there is some type of infection, and this is something that, is just kind of starting to be looked at again in small pockets, but is there potential for some type of phage therapy of Crohn's disease if there's an infection involved?
Yeah, I think that any way to kill a bacteria would be beneficial if the bacteria is harmful. I'm not sure that phages are the easiest way to prove it, because phages are very kind of boutique, and you have to find the phages that work against this bacteria in this person. So, if you wanted to prove that tuberculosis germ theory, I think you'd find it easier to use pills that are available at your pharmacy than phages. If you want to cure somebody with a devastating Mycobacterium abscessus infection that's resistant to everything, then you would say, let's try phages, because we have no other opportunities.
Okay, and the last question I have, and then Annabelle and Angela, feel free to ask any others, is about this concept of autoimmunity. Years ago you did, and this is how I actually found you initially, and then we were connected and had some amazing conversations. But you did a presentation about autoimmunity, asking the question, is Crohn's disease autoimmune? And at that time, and this was a number of years ago now, at that time, autoimmunity was thrown around a lot in the literature and in presentations when people talked about Crohn's disease. I think it's been walked back a little bit. I think most of the time now people say immune mediated, although we still hear autoimmunity as well, particularly when talking with immunologists. And, there are some biomarkers that have been identified years in advance for Crohn's disease, like ASCA is one that's more prevalent. There was a recent one, I believe the anti-GMCSF, I said those letters probably in the wrong order, but so there are these biomarkers. People wonder about autoimmunity. What are your thoughts about the evidence around whether Crohn's is or is not autoimmune?
Well, when I wrote an article around it 12 or so years ago, I was afraid that by now somebody would have said, here are the autoantibodies, you were wrong. The ASCA, the anti-Saccharomyces cerevisiae antibodies, is not autoimmune. It would be xenoimmune, if you like, or I guess xenoantibodies, because it's against microbial products. It's not against host products. I did read the article from the Toronto group on
Dr. Murtha, I believe.
Dr. Murtha, and that's really fascinating. But anti-GMCSF or anti-interferon lead to immune deficiency. So then it kind of brings a kind of a new circularity in that there could be autoantibodies against the part of our immune response that makes us deficient to how we handle microbes. So that part I think is quite plausible. And then we would get in probably to a more semantic argument about what is autoimmunity. But the idea that at age, 27 or 37, my body just decides it doesn't like me and attacks me without some sort of an external provocation, I find has not been very persuasively shown in the case of Crohn's disease. And even in other immune-mediated diseases, inflammatory diseases, I think external triggers are increasingly being searched for, as in the example of EBV and MS that you cited earlier.
Great, thank you. Annabelle, Angela, do you have any questions?
I do. Thank you, Dr. Behr. That was all really interesting and gives us a lot to think about. I have a question that's probably basic and it may have been addressed, but just, I might have missed it, but just from your observation, what is your opinion as to whether infection plays a role in a subset of Crohn's patients or perhaps the majority of patients? What is your feeling on that?
Yeah, I don't know. It comes down to Ildiko's point about having a reliable test. When we looked with microscopy and surgical resections, it was the majority, but that hasn't been repeated by anybody. In terms of culture, though, it has been a very small number of humans that their sample has given a positive culture for Mycobacterium Avium Paratuberculosis. So that remains sort of an unanswered question.
Okay. And I was also very interested by your comment that perhaps we calibrate success differently in terms of IBD and in what we see in trials and effects. Can you elaborate more on why that may be so compared to some other things that are studied? I thought that was interesting.
I honestly don't know. I don't work with regulators. In the world of tuberculosis, I know that we spend a lot of time discussing what's the endpoint of a vaccine trial. And so, you know, what I've learned over time, by the way, is just simply if I don't know the answer, I move the question because I'm a full professor and I have that prerogative. So let's take a vaccine for TB. And you bring doctors into the room and say, what's the endpoint? And they're going to say symptomatic disease. We want people to have less illness. You bring public health people into the room and they'll say microbiologic positive. We don't want to have onward transmission. And this came up with COVID as well, right? So our vaccines were licensed for PCR positive symptomatic people. But somebody else could have said, well, actually, I don't really care if they're symptomatic. I'd be happy if we'd reduce by 93% the amount of asymptomatic shedding as well. That would be a public health goal. But if your goal is to keep people out of the ICU, you'd say, well, no, I'm going to license it to reduce the number of ICU visits. So just by the people you bring into a room, the definition of success for something we know really well can change. And then you can start to get into these arguments. And so that's for something we know the cause, we know the consequence, we know the outcome. We've known about this since 1882. I wonder if in immune-mediated inflammatory diseases where we don't know the cause, where we have more hypotheses and more theories and more explanations, it gets harder to come up with a single simple definition. And so you end up with scores and scoring systems, and you go down this much and down this much, and you look for an early win, you look for a late win, and at the end, you know, if you find any win early and any win late, you're doing better than nothing. But you don't have the same precision as saying, we got rid of the SARS-CoV-2, or we got rid of the Mycobacterium tuberculosis. But you'd have to talk to people in GI about this. I'm not sure if the early remission, late remission is unique to GI. I think this may also be seen in rheumatology.
Thank you.
So how about using bacteriophage for testing?
For testing what?
For testing for, say, MAP, is it, or even for TB, is that even a possibility?
Yeah, there's a company in England that's developing a phage-based diagnostic platform. I don't know if it's been approved by EMA or by the FDA yet. And we don't yet have all the information on whether it's more sensitive than culture and if it is equally specific to culture. So I'm aware of a group developing this work, but I'm not sure yet if it's at or about to come into the primetime arena.
Okay.
And I guess lastly, is there anything you wanted to tell us about your current research, any research you're doing now or that you're hoping to do on these topics?
Yeah, so having seen that 2 independent randomized controlled trials showed a benefit of a ad hoc cocktail of antibiotics, I thought, well, why don't we at least go to the lab and try combining antibiotics in a rational way and see if we can do better. And so right now, one of the things we're doing is just going to try a whole bunch of different antimycobacterials. And if what has been given to people is optimal, then at least we'll know that. But if we can find a combination that's better, then I'd be interested in seeing if we could advance it a few more steps along the way.
I think that would be amazing and very high value research. And again, it's hard to sometimes get the answers on these older drugs like antibiotics. And I'm so glad that you're doing it. You know, what I've always said that I so appreciate about you, among many things, is that you're a real scientist. You know, you don't say, you're very careful in your choice of words about what the evidence shows. You're very careful in your methods. You're very cautious about how you read studies and other people's methods. And I really, really appreciate your scientific precision when you're doing this work. So I hope that you are able to do that work. And I think that would be really very value added. And I hope that the GI and IBD community support that research, because I think it's really, really important research.
Well, those are very kind words. Thank you very much.
So thank you for being here with us today. I really, really appreciate it. I did want to, before we sign off, just give a brief shout out to the Human Para Foundation that Annabelle and I know well, who does, I think, has rightly focused in on MAP and trying to advance the science of MAP over many, many years. And I think I'm grateful that somebody's doing that and that they're doing that. So I just want to give them a shout out. I know this topic is of great interest to them. So I want to thank you for all your work over the years. I think very thoughtful work, and also for joining us today. I really appreciate it. I know you're not somebody who gives, lots of interviews. So I really appreciate you taking the time to be with us today, Dr. Behr.
Great. Thank you for having me. Have a good day.